Okay, so Dr. Robert Guralnick is Associate Curator of Biodiversity Informatics at the Department of Natural History and the Florida Museum of Natural History. His research is focused on the causes of spatial temporal changes in genetics and species diversity. His talk today is entitled Species Distribution Modeling. Let's welcome Dr. Guralnick.
Yeah. Thanks, guys. I have both red and green pointers here for a little bit of color here. I think I'll probably just stick with the green. And what I want to do here is a little different than what other presenters have done. And it's basically to give you a kind of a conceptual framework and background in the topic of species distribution modeling. And I'm going to be a little lighter on empirical examples and a lot more focused on conceptual ways that we think about doing species distribution or niche modeling. You've heard a little bit about this from Janine and others regarding how we basically create habitat models. What I want to do is walk you through some of that theory and a little bit of the practice in order to kind of set the stage, hopefully, for a lot of the discussions we're going to have in the next week. And so I apologize in advance for being light on empirical and more kind of driving on concept and what we're doing when we actually do niche models. You know, kind of think about this as almost like a niche modeling introduction seminar, mostly focused on modeling frameworks and how we do the modeling. All right, so with that introduction, in regards to conceptual background, what I want to focus on here is the conceptual basis behind species distribution modeling. So I'll use the term SDMs for that. I'll also use the term ENMs, ecological niche models, and those are not entirely and completely synonymous, but they're close in proximity to each other. And I think the argument could be made that when we talk about the difference between an ecological niche model and a species distribution model, in order to do species distribution modeling, we really have to have some information about the ecological niche, but SDMs can incorporate more about movement biology, movement factors for organisms, like dispersal and colonization parameters. And I also mentioned that niche modeling, excuse the little typo here for us, but is really involving transferring from sort of thinking about geographic space where species are found, the locations of individuals and populations across their ranges, to environmental spaces where we can think about niche characteristics, physiological tolerances, and those kinds of characteristics. All right, with that in place, what I want to do is just step back a second. And I know there are a bunch of people in the room who are not directly from the ecological realm. How many of you are familiar with the sort of core concepts and niches regarding Grinnellian, Ottonian, Hutchinsonian niches? Raise your hands up so I can see it. So there are some people in the audience who haven't had that background and kind of conceptual frame for thinking about these definitions of what we mean by niche. And so I'll just give you some of the key concepts regarding what we mean when we say Grinnellian niche or Ottonian niche or Hutchinsonian niche. And Joseph Grinnell was actually a California naturalist, very well-known ecologist, evolutionary biologist. And studying the California thrasher, he wrote a really seminal paper, and that's a little California thrasher silhouette. You can see here in the chaparral in Southern California. But what he noticed was that the thrasher's behaviors in the chaparral and its sort of tie between the habitats and its behavioral specializations were really tight. And from that, Grinnell conceptualized the idea of this Grinnellian niche, which are really the habitat requirements, the abiotic, non-consumable characteristics that are important for fostering persistence. Charles Elton was one of the most famous ecologists of a generation. And Elton was thinking more about consumable resources and kind of the differential relationships between trophic interactions, predator-prey dynamics, and his concept of the niche really stemmed from realizing that in different systems across the globe, you could see different predator-prey dynamics in terms of major predator species like predatory birds and their small rodent prey that looked very similar in different areas, even though they're different species performing those actions in those different areas. And so the Eltonian niche is really about these biotic interactions, and it's typically considered at local scales. And then J. von Hutchinson and the Hutchinsonian niche really considered niche as an n-dimensional hypervolume defining both these environmental conditions and resources, and made this critical distinguishing factor between the fundamental and the realized niche. And so regarding that distinction between the fundamental niche and the realized niche, and that's actually relatively important for what you're modeling when you're doing species distribution modeling or niche modeling, you can think of the fundamental niche as that full range of environmental conditions, biological and physical, under which an organism can exist. And because of biotic interactions, competition, and predation, the realized niche is sort of a subset of that fundamental niche. So that's something that most of you probably had in in ecology classes from hopefully introductory classes. So the abiotic requirements in the Guralnick niche are these non-consumable resources like climate or geophysical characteristics, substrate, or nutrients, right? Okay. And those biotic requirements in thinking about the Altonian niche are really interactions driven with other species relating to competition, predation, exploitation, commensalism, and mutualisms. All right. So when we think about the fundamental niche, I just want to point out that that's really intimately tied to thinking about individual population species physiological tolerances, and we can think about the optimum range for a population to persist across the environment, where that would support population growth. And the optimal range over here is a sort of key part of the fundamental niche, and you may have these tolerance areas in the space of environmental variables where taxon can persist but not necessarily grow, or we can consider those more maybe as sink areas across geographic space. And then there are these areas, of course, where taxa just can't survive. And of course, that just to point it out again, this realized niche is really often, but not always, a subset of this fundamental niche in niche space when we're thinking about these key niche parameters. Okay. So really important here is also thinking about movement. And when we think about sort of the geographic components in niche space, movement factors are really critical, and these are often historical biogeographical considerations. And what I want to point out here is just a really simple fact that where species are found are often driven by these historical processes, evolutionary processes, that may mean that a species could be found in many areas that are abiotically and biotically suitable, but just simply can't get there because of these dispersal limitations or other features of colonization histories of taxa. And so we have to think about movement a lot when we're thinking about trying to model species distributions geographically. So putting this all together, this is a really, I think, sort of conceptually nice framing around what we're trying to model and how we're trying to generate habitat models using especially presence-only data. And this is really about, why are species found where they're found, and these key limiting factors, including these biotic factors shown as B in the sort of BAM diagram, abiotic factors, and movement factors. And it's the area of overlap of these biotic, abiotic, and movement factors that are critical for determining the occupied distributional area and the overall potential distributional area, which is sort of this intersection of the abiotic and biotic factors. Okay, so putting this all together and kind of thinking more about this, each species is going to have different ways that those abiotic, biotic, and movement factors play out over their evolutionary histories. So these overlaps are often almost always different and actually very hard to know a priori. So in some cases, we may have strong biotic interactions, strong abiotic factors that limit distributions, and strong migration factors that limit distributions, causing the occupied distribution to be very small. And in other cases, we may have a wide-ranging species with relatively broad abiotic characteristics and biotic interactions. That means that this is a very different shape of what we consider to be the occupied distribution of a taxon. And those really matter when you're trying to basically do species distribution or ecological niche modeling. So to kind of come back to this and just cement this a little bit, the occupied distributional area for a taxon is really this intersection of the abiotic, biotic, and movement limitations, shown as G0 here. This GI, this invadable distributional area, is really simple to get your head around. Basically, this is the area that could be invaded. These are areas with suitable abiotic and biotic conditions, but the taxon has movement limitations that disallow it from accessing those invadable areas. One interesting thing about Charles Elton was he was also the father of invasion biology, and really thinking about invasions and this question about abiotic and biotic conditions where invasions can happen is really crucial. And that full area in total, when you union the occupied geographic areas and the invadable geographical areas, is this GP, the potential distributional area. Now, the core of niche modeling, I kind of alluded to at the beginning of this, is really about translating between geographic spaces and environmental spaces. And there's this sort of function, this eta g, that takes this geographical components of where species might be found, shown across North and South America with these black dots representing point presences, and you can translate this into a multivariate space of environmental dimensions associated with G, and you can do an inverse relationship where you can reproject this this environmental space back onto geography, where these geographic locations correspond to environmental combinations of that environment. And so a lot of niche modeling is really being able to move across these spaces and being able to model across these spaces in terms of geographic and environmental space. And because of that, there are reflections of the geographical components of these BAM diagrams in environmental space. So the potential environmental niche is the eta GP, which is again the eta GO and the eta GI, which sums up to the union of EO and EI. And this is basically reflecting these relationships between the geographic space and the environmental space. And at heart, the key fundamental thing we're modeling when we model niches using especially these correlational approaches is really what's called the existing fundamental niche. And this is actually an intersection of the true fundamental niche conditioned by the fact that there's a subset of conditions on a real-world landscape where a taxon could be found, which means that the fundamental niche is usually actually broader than this existing fundamental niche shown here, which is a key thing we're going to be wanting to measure. Okay, so that covers a bit of the conceptual groundings of niche modeling. Let me talk about what we do here. So how do we model this existing fundamental niche? And there are multiple methods to do this. There are different classes of models that people apply to these methods. I'm going to cover two classes that are very well known and well used, one of which is this mechanistic approach to assembling knowledge about the fundamental niche and species distribution models, and this more correlative approach that is used very, very commonly. So mechanistic models are basically ways to experimentally determine a species' fundamental niche. These can be done in growth chambers, looking at physiological tolerances, or using reciprocal transplant experiments, or they could actually be generated from biophysical models where we know something about heat loss functions and thermobiology to basically be able to generate knowledge about how species function biophysically in their environment. And you can also derive these from known physiological data from the literature. And then with that, those knowledge about physiological tolerances and mechanisms in place, you can simply union that with the environmental conditions for a population or a species. This other class of models is the one that I'll focus on, which are these correlative models. And that really uses data about species presences and environmental covariates to try to estimate that existing fundamental niche. Okay, so mechanistic models are really useful. They're kind of the gold standard in a sense. They're experimentally determined. These generate response curves and, again, focus on physiological performance, mortality, and reproductive success. And that does give you effectively more of the fundamental niche as opposed to the existing fundamental niche. But that's very, very useful. But the problem is that these data are hard to come by, especially for non-model organisms. And then there's questions about the effort that has to go into basically generating the data needed to get a handle on these physiological tolerances and the relationship to the fundamental niche across, and especially at the broad scale across ranges. That can be difficult given that there's microevolutionary, macroevolutionary forces are operating on different ways across different portions of a species range. The approach most commonly used in terms of species distribution modeling are these correlative niche modeling approaches. This relies on species presence data plus a set of environmental covariates, and is really about transforming from that knowledge about species presence and the covariates, the conditions where that species was found, to be able to translate from that geographic space to that environmental space. And then we want to basically transform back to our geographic space for species distribution modelings, either using interpolation approaches, or often we're transferring models from where we sample the data to areas where we didn't sample in order to look at things like invadable ranges or potential responses of species distributions under climate change. And the goal is typically predictive for areas that aren't sampled, and the output is often discussed as being habitat suitability. All right. So about those environmental covariates for niche modeling, that's where the remote sensing data comes in. Really any environmental covariate that is summarized by a raster, these are often typically climate variables or biophysical variables, geological variables. They can be continuous or categorical. So these are really just traditional typical environmental covariates. These include often altitude and bathymetry, slope and aspect, soil characteristics, climate. Those are the common ones that are used, but there's certainly all ways that one can bring together the right environmental covariates. And really, the environmental covariates that you choose should be really based on what you know about the species. So species may have differential factors that are critical for determining their environmental niche, and it's up to the modelers to really know the biology of their organisms well enough to make the right choices about what environmental covariates are actually predictive of their niche. Okay, so in sum, what we're doing here is we're basically collecting presence points from the environment. We're selecting an area where we think that is accessible for that species, and that's shown in this box. We're projecting that into e-space, looking at a model of niche in those environmental dimensions, and we can then project those back onto the same landscape, or we can transfer them to past or future or different areas where those have been sampled with attendant challenges about these transfer functions and being able to actually do interpolation versus extrapolation properly. And that's kind of the heart of the idea behind correlative niche models. Let me give you an example of how this works in terms of thinking about some of these questions regarding transfer of models from one spot to another. And this is kind of a classic example, and it's one that I think is elucidative of the challenge here. And so this is an example using the Asian tiger mosquito. This is native to Southeast Asia and India, and it's spread throughout the US over the last 30 years, 35 years. And what we can do is we can model Aedes albopictus distributions and using point presence data in its native range. So this is the native range of Aedes albopictus. This is actually the model distribution based on these point presence records and environmental covariates. This is obviously an important taxon for its disease risk in regards to dengue. And then from that, we can actually predict, we can take the niche model and reproject it into areas that are in where Aedes albopictus has invaded. There's an invasion front, and so we know that these are non-equilibrium dynamics. We can still model where that might occur in, say, the US based on projecting that niche model onto this geographic or this climate landscape in the conterminous US. And then we can actually ask, if the models are actually supported based on the data of the invasion front for Aedes albopictus. And here's that diagram showing you how Aedes albopictus has spread across the US at least over the last 30 years, matching relatively well what we see from this prediction from the niche modeling. Make sense? Okay. So and then there's a niche model showing you that, and you can see that there are cases where the niche model is clearly wrong. These are areas where the model is not predicting presence, but we see presences of Aedes. What's kind of nice is that we can also model distributions under future climate conditions. This is projecting models into the future. There are all kinds of attendant challenges about doing this regarding extrapolation, but when we do that, we can actually see what Aedes albopictus distributions might look like under IPCC A1B model predictions for climate change, right? That's useful. Okay, so so far I've focused this purely on the framework of thinking about these sort of models around species presence and not around thinking about absence. Absences are really critical for documenting change dynamics and for getting away from thinking about kind of potential distributions and fundamental niches towards more realized distributions, which I think is really critical. They are probabilistic, unlike presences, and there's a whole class of modeling in the framework of occupancy modeling that is focused on detection probabilities and how to simultaneously assess detection and site occupancy. Those occupancy models are clearly connected to this larger world of species distribution models. I don't want to go into too much detail here, but I think at the boundaries between occupancy models and species distribution models and using them together, we're able to do a lot to be able to detect change dynamics where species are changing across time and space based on bringing together these presence data with these presence-absence data, typically from more of a monitoring framework. So how do we assemble absence data? Most of our data that we're using for niche modeling, I think that all the niche modelers in the room primarily use presence-only data. most of the time. But we do have growing bodies of absence data. These presence data are based on incidental records. They don't really have a taxonomic scope, where there's no sample method usually described. But these absence data can be generated from more of an inventory approach, where we're looking at elementary or extended or summary inventories at broader spatial and temporal scopes. And those can generate these key absence records that can feed into models. And again, I would say that the reason to care about absences is that if we want to understand realized distributions and how those change, it's really critical to bring in those absence records. And absences actually can be used in model construction. They can also be used in model testing frameworks. What we're going to focus on here for the rest of this time is just a little more on these presence-only models. So how do we do this in practice? In niche modeling, what we often do is collect presence records, but we have to also define what takes the place of absence in these models, and typically that's either pseudo-absence or background. These are basically the same idea. It's drawn from the climate conditions in this accessible area. Pseudo-absences are records from that accessible area that haven't been sampled as presences. Background is just basically the whole of those values of environment from that accessible area, and it really doesn't matter unless you have a really small area that you're sampling from. So just a key point that when we do these kinds of models, the spatial bias and point presences is really a problem, since if you draw a bias sample of the occupied geographical area, it also means you have a biased occupied environmental space, which is bad. And there are approaches out there that help with trying to fit these sample biases when you're modeling. One thing that you can do is you can actually quantify these sampling biases. So this is actually a paper from Karsten Meyer in 2015 that I was involved in. And the idea here was to plot point presences shown in these sort of darker colors against the known range maps of the species, and then use random sample points across these ranges to calculate something about range coverage and geographical bias through a null modeling approach. What you can kind of see here in kind of a classic example is here's a taxon that has a very wide range across Siberia into eastern Russia. Most of the point records are drawn entirely from Western Europe. So if you use these points to try to make a distribution model, you have all kinds of challenges about strong geographic biases feeding into those models. So I'm going to cover really quickly here just how people think about the modeling tools that are used. We can think about presence-only algorithms, and these are very simple models that don't use anything about those background or absence points. They basically simply infer either an envelope rectilinearly or a hole, and this whole region of E-space is then used to project back into geographic space. So everything inside the envelope is suitable and everything outside is not. These are very simple. One of the key tools that people use a lot, and although there are many, many tools out there that are all based on sort of a form of GLIM or GAM model, but one of the key ones that people still tend to use is Maxent, which performs relatively well across a variety of modeling scenarios. It's basically trying to maximize sort of smoothness in geographic space while minimizing dispersiveness in covariance space using an iterative approach. And it's basically uses an iterative approach to start with a fully uniform distribution over all the grid cells, and then basically tries to maximize gain using a likelihood statistic with trying to get the best probability of presences given the input data in relation to the background, which leads to this final probability distribution from which you can generate variable coefficients for defining probability of presence. And then finally, there are a couple different approaches for doing ensemble models where you can use multiple modeling methods, which are often used in climatological research, to basically generate a summary model that takes into account the uncertainties from different modeling approaches. And so there's a great toolkit called Biomod Tool 2 that is used for these ensemble approaches. So in this case, you can combine model predictions. There are many ways to do these combinations, but you can get the best model results from all the model types, or potentially get the worst model results from all modeling types. So that's a key way to think about how to move into the framework of actually doing these species distribution models. I want to do a time check of about 5 minutes. And so what I'm going to do for a couple minutes is just introduce very briefly the concept of evaluating models. And then I'll hopefully have a couple minutes for questions. if you have them. So the key way that people think about validation, and the best way to do this for sure, would be to create your model and then go sample in the field and get more presence data, and also try to get non-detection data to see if your model is valid. But that's not how people typically proceed. I don't know if there are many examples at all in the literature of people going out and actually verifying their niche models back in the field. Most people do is stratify their data using randomized subsetting to look at internal data sets where you have a training data set to train the model and then a testing data set to see how the model performs. But there are multiple ways to do this, including cross-validation and partitioning, where you can partition data sets into multiple subsets and then test them across those subsets. And then use the non-test partitions in combination for model calibration, model training. And then for background points, just a note that Maxent treats them as pseudo-absences for the purposes of these post-calibration evaluations. So this whole basis for model validation is really built on sensitivity and specificity, the true positive and false positive rates, and the true negative and false negative rates, which are used across remote sensing. So I don't think I need to belabor this point a whole lot. But there are different ways that people do this. They use threshold-dependent metrics like Cohn's kappa, or the true skill statistic, which is really looking at the hit rates versus the false positive rates. These often require producing binary maps for this. And then there are threshold-independent metrics like the area under the curve. Regarding thresholding, this is one of the points of niche modeling and species distribution modeling that is almost an art. This is trying to determine basically from a continuous probability surface that you have here, ranging from 0 to 1, where you can see 0 is low suitability, 1 is high suitability. From this, you can basically choose a threshold in order to determine what you consider to be ultimately presence or absence for a single outputted species distribution model. And those thresholds vary. And the way that people typically do this is simply by trying to balance the commission and omission errors across the models using these outputs about sensitivity and specificity. So looking at the time and being cognizant of that, there are a couple more things I could mention regarding the use of these area under the curve methods for looking at niche models. But what I want to do is just quickly mention that, and in order to have a couple minutes for questions, that the key thing I want to mention is that one of the important parts of the area under the curve thinking is that you can certainly get your area under the curve for your receiver operating curve, but the key thing that I think is really important is not so much looking at these values, since they're spatially autocorrelated, not entirely independent. One critical thing in the modeling frameworks is to think about how well your area under the curve looks for your training data set and your testing data set both, so you can verify whether your model is over or under fit. And so that's a key way, I think, to use these model validation tools properly, because there's a tendency for models that use a lot of parameterizations to be highly overfit to the data. And so looking to see whether your test AUCs are relatively similar to your training AUCs is really important. With that, I see the time ticking down, so I think I'll leave it there for where we've come to. I'll thank you for your attention and be happy to take a couple quick questions. And I hope the overview is useful as we go into these next sessions over the next week to kind of talk about how we'll bring together the in situ data, the presence absence data, with the remote sensing data and be able to integrate more effectively across them. So with that, thanks a lot. Appreciate your attention.